I'm the director of the Stanford Cancer Institute, and this is our most impactful seminar series for bringing leaders in cancer to Stanford to learn about their breakthroughs in cancer. Now it's my pleasure to introduce Dr. Bill Kaelin. So Dr. Kaelin was born in the New York City suburbs and raised there. He attended Duke, where he double majored in math and chemistry, followed by getting his MD at Duke. Then he was a house officer at Johns Hopkins and a chief resident there, followed by a fellowship in medical oncology at Dana-Farber, and then did a postdoctoral fellow with the late David Livingston, a legend in cancer research at Dana-Farber. He's had a spectacular career, is in the National Academy of Sciences, National Academy of Medicine. He won the 2019 Nobel Prize along with Peter Radcliffe and Greg Semanza for his groundbreaking discoveries in oxygen sensing. He serves on the Board of Trustees of Duke and the Damon Runyon Cancer Research Foundation. And just a few personal notes. I was a fellow at Dana-Farber where I got to know Dr. Kaelin when he was, I would say, maybe in the early stages of revealing the fundamental mechanisms of von Hippel-Lindau syndrome, which is a tumor suppressor gene and cancer predisposition syndrome. And I think we all appreciated Bill at that point as having a keen intellect and being very laser focused on mechanism. I did my postdoctoral fellowship on the same floor in a building called the Mayer Building. It was an incredibly dynamic floor filled with postdocs and exciting discoveries, but none so as exciting as the discoveries made in Dr. Kaelin's laboratory at that time. And I would also say that I remember him when he was clinically active as an oncologist. We had something at Dana-Farber called Doc of the Day. And this is when fellows were maybe more lightly scrutinized than they are now. And so there was basically a fellowship clinic where patients would come for their follow-ups. And there was on the blackboard written Doc of the Day. That was the attending doctor. And it said, Bill Kaelin and his pager number. And so one day I was in the clinic. You may not remember this, but I was in the clinic and Bill was Doc of the Day. And I had the pleasure of paging him to come see a patient with me. And he was an excellent clinician at that time as well. So it's just been a pleasure to have followed your career, Bill, over the years, and incredibly proud of you for how you've been recognized and the discoveries that you've made. So I'd like to welcome to the stage now Dr. Bill Kaelin.
Okay. Well, thank you, Steve, for that very lovely introduction. I try to manage my travel schedule because it can get out of hand pretty quickly these days, but this is such an incredibly special place. I hope none of you take it for granted. This is such a special place. It's really a privilege to come share some time with you today. As was mentioned, I went to Duke. When I was at Duke, I never envisioned that one day Stanford would be part of the Atlantic Coast Conference, but on the other hand, we're seeing lots of things this past few weeks that I never thought I would see also. So let's put aside some of the craziness outside in the world and spend a little time together. Now, what I thought I would do is spend about 10 minutes for the benefit of the students doing some background review, so my colleagues will be forgiven if they want to answer text messages and check emails for the first 10 minutes, and then I'll share a couple unpublished stories with you. So here's the obligatory disclosure slide, and I'll try to mention the most relevant ones as I go along. And here are the people who've worked on von Hippel-Lindau disease in my lab over the years, and I highlight in red one of your colleagues, probably known to many of you, Dimitrios Kalivas. And here's a partial list of the collaborators who have helped us with some of the more recent work that I'm going to be sharing with you. So my lab's had a longstanding interest in von Hippel-Lindau disease. This is presents as a hereditary cancer syndrome. It affects about 1 in 35,000 people. It was first described in the medical literature about 100 years ago, but in hindsight, some families have had this disease for centuries. And the tumors seen in this disease include the most common form of kidney cancer, which is called clear cell renal cell carcinoma, blood vessel tumors of the brain, eye, and spinal cord called hemangioblastomas, and neurocrest tumors called paragangliomas, which when they arise in the adrenal gland are referred to as pheochromocytomas. Now, for the students, you would be forgiven if you said this looks like an autosomal dominant disorder, but in fact, like most hereditary cancer syndromes, it's actually caused by a recessive mutation, in this case in the von Hippel-Lindau tumor suppressor gene. And unfortunately, if you're born with a defective copy of the VHL gene, you have a 90% chance that a susceptible cell in your body, such as in the kidney or the brain or the eye, will spontaneously lose or mutate the remaining wild-type allele, and that's why you have this sort of pseudo-autosomal dominant pattern of transmission. And as you would predict from the knowledge that germline VHL mutations predispose to, for example, kidney cancer, again, clear cell renal cell carcinoma specifically. Now, if you look at non-hereditary clear cell renal cell carcinomas, you again see that typically both the maternal and the paternal copies of the VHL gene have been mutated or lost. But here, both mutations or hits, if you will, have occurred sometime after conception, in contrast to VHL disease, where the first hit has occurred in the germline. Now, I knew from my clinical training that was just alluded to, and thank you for not throwing me under the bus with terrible stories of my clinical capabilities. I knew from my clinical training that the tumors linked to VHL disease or seen in VHL disease are highly angiogenic, and that's because they overproduce vascular endothelial growth factor. And on occasion, these tumors also cause patients to make too many red blood cells, and that's because they sometimes ectopically produce erythropoietin. And what VEGF and erythropoietin have in common is that they're normally induced when cells or tissues are not getting enough oxygen. So this was the clue that maybe by studying the VHL gene and its protein product, we would learn something about oxygen sensing, because it looked like oxygen sensing had been hijacked or corrupted in these tumors. Now, we knew from the work of multiple labs, including my fellow laureates, Greg Semenza and Peter Ratcliffe, that many of these so-called hypoxia-inducible genes, such as VEGF and erythropoietin, are under the control of a heterodimeric transcription factor called hypoxia-inducible factor, or HIF. And we also knew from their work that the alpha subunit is normally degraded when oxygen is plentiful, hence hypoxia-inducible factor. And over the course of time, our lab and others showed that the VHL protein product is part of a ubiquitin ligase that, in the presence of oxygen, binds directly to the HIF-alpha subunits and targets them for proteasomal degradation, whereas when oxygen levels are low, or the VHL protein has been mutated, now HIF-alpha can accumulate, dimerize with its partner protein, HIF-beta, or alternatively called ARNT, and activate genes such as VEGF. So this explained perhaps the deregulation of hypoxia-inducible genes in tumors lacking the VHL protein, but it begged the question, how does the VHL protein know, if you will, whether oxygen is or is not available, and hence whether it should or should not target HIF-alpha for destruction? And in work that we did and that Sir Peter Ratcliffe did, working independently and in parallel, we showed that in the presence of oxygen, one of two proallele residues in HIF-alpha gets hydroxylated, and this then creates a high-affinity VHL binding site. And I have to say, at the time we were doing this work, there were a lot of complicated, baroque, sometimes even contradictory models for how oxygen would be sensed. So one thing we had going for us when we made this discovery is even I could see how this would be linked to oxygen availability because of this oxygen atom here. So this was very satisfying. And then work that Peter Ratcliffe did, Rick Bruick and Steve McKnight did, and also work from our lab showed that the enzymes that do the work here are the EGLN, or also called PHD, prolyl hydroxylases. They split molecular oxygen, and they use one of the oxygen atoms to hydroxylate HIF-alpha, and again, as I said, that generates a VHL binding site. These enzymes also require reduced iron, and that explains why if you add iron chelators to cells in culture, you will stabilize HIF and mimic a hypoxic response. And they also require a cofactor from the Krebs cycle, which is variably called 2-oxyglutarate or alpha-ketoglutarate, and 2-oxyglutarate gets decarboxylated to succinate during the hydroxylation reaction. And although I'm not going to talk about it today, a number of pharmaceutical companies quickly figured out that you could make 2-OG competitive small molecules that had drug-like properties that would inactivate these enzymes and basically mimic a hypoxic response and cause people to start, for example, activating genes dedicated to red blood cell production, such as erythropoiesis, and these are now used in many countries around the world for the treatment of anemia. For example, there's a drug called daprodustat that's now approved in the U.S. for the treatment of anemia linked to chronic kidney disease. And I've been involved with a drug called roxadustat that's been approved in 44 countries around the world and is being taken by millions of patients to treat anemia. Now, these enzymes I just introduced to you belong to a larger superfamily of so-called 2-oxyglutarate-dependent dioxygenases. So here are the 3 members of the prolyl hydroxylase family that modify HIF. And I would argue you might want to have at least a passing knowledge of this superfamily for a couple of reasons. First of all, they also include the so-called Jumanji C histone demethylase. And can you see this pointer better than I can? Because it's a little faint for me, but you're okay. So it includes the Jumanji histone demethylases and also the TET enzymes involved in DNA demethylation. And you may know that a number of so-called oncometabolites, such as succinate and 2-hydroxyglutarate, are thought to transform cells by interfering with the function of specific enzymes in this family. In fact, if I was giving another 1-hour research seminar, we could just talk about that for an hour. But we're going to keep this at 40,000 feet for the moment. Now, another ingredient that I didn't share with you is the other reason these enzymes, these prolyl hydroxylases, are so important for oxygen sensing is they have very low oxygen affinities, or put another way, they have very high oxygen KMs. And as a result, the enzymes that modify HIF are exquisitely sensitive to changes in oxygen availability in a physiologically relevant range. This is in stark contrast, however, to the collagen prolyl hydroxylases that you might have learned about in college biochemistry. So these enzymes have extremely high oxygen affinities. You'd have to be virtually anoxic before the collagen prolyl hydroxylases would become inactive. So we became intrigued a number of years ago with the question of whether any of the other enzymes in this superfamily might also have the potential to be oxygen sensors. And we honed in on the histone demethylases, the so-called Jumanji C histone demethylases. And I'll remind you that it was Yi Zhang, who was then at UNC, who first taught us that these were histone demethylases. What they do is they hydroxylate, in this case, the methyl group of the histone, which is then unstable and given off as formaldehyde. So we thought it was important to understand whether the histone demethylases were more like the enzymes that modify HIF, and that is to say potential oxygen sensors, or were they more like the collagen prolyl hydroxylases and effectively oxygen insensitive? And the short answer is we could find many examples of both. So just let's focus for today on KDM6A, which is also called UTX, parenthetically a tumor suppressor. So this is an enzyme that removes the usually repressive H3K27 trimethyl mark. So we collaborated with Pepi Koivunen and her team at the University of Oulu. So these are real card-carrying, like cold room enzymologists, biochemists, and they helped us characterize this enzyme with respect to oxygen affinity. So if you look at KDM6A, it has a very high oxygen KM, or a low oxygen affinity. In fact, it's very close in that regard to the enzymes that modify HIF. But if you look at the paralog KDM6B, so this is the other member of the family that demethylates the trimethyl mark on H3K27, it has an extremely low oxygen KM, or a very high oxygen affinity. And this is what we see with a lot of the histone demethylases, that there's usually at least one member of the paralog family that's an oxygen sensor. And since this is published, I'll just tell you that we were able to show that some of the effects that have been linked to hypoxia over the years in cell culture actually in some cases appear to be directly through changes in this histone demethylase rather than through the activation of HIP. And so I think this is another way that oxygen can affect cell behavior, and it's made us think about the role of hypoxia in the stem cell niches during intrauterine development, which is also hypoxic in mammals, as well as within solid tumors. Now, I showed you this a moment ago, so we knew that loss of VHL leads to upregulation of HIF. So that's the correlation, but I always remind students, don't immediately make the leap from correlation to causation. It was plausible that HIF was the problem with respect to these tumors, but someone had to do sort of what we call the necessity and sufficiency experiments. And so years ago, we had shown that restoring the function of the VHL protein in a VHL null renal carcinoma cell line would suppress the ability of these cells to form tumors in nude mice. And so Keiichi Kondo in the lab took these cells and introduced a form of HIF-alpha, and in particular HIF-2-alpha, that's no longer recognized by VHL because the proline hydroxylation sites have been converted to alanine. And now these cells once again could form tumors. And in a complementary set of experiments, Keiichi showed that you could eliminate HIF-2-alpha then with short hairpin RNA technology, and these cells would lose the ability to form tumors. And much to our surprise, when we did analogous experiments with HIF-1-alpha, the more famous canonical member of the family, if anything, we saw the opposite effect. So this really honed our interest on HIF-2 as being the culprit. In fact, in many human kidney tumors, you can see that HIF-1-alpha is silenced, and they're being driven exclusively by HIF-2. Now, for the students, not shown here are any of the controls, right? And one of the things I learned from David Livingston is like controls, controls, controls. So your required reading is this paper where I tried to capture in about 8 to 12 pages every mistake and artifact I think I've seen in the last 25 years ago. Sadly, I'm sure we committed some of them ourselves. But I wrote this as a tribute to my late wife who died of cancer. You may know that our friends in the pharmaceutical industry tell us that 50 to 80% of the time when they go to replicate our findings with respect to new targets, the findings either can't be reproduced or they're not robust, meaning they're only true under extremely narrow conditions and are unlikely to be the foundation for a meaningful drug discovery program. So if you're going to do this, if you feel compelled to do target validation studies, maybe reviewer 3 is demanding a target validation study, please read this paper first because there's a lot of nonsense in the literature and we have to do a little bit better when it comes to target validation. Now, what could we do about this? Well, fortunately, by the 90s, a number of pharmaceutical companies were making inhibitors of VEGF. And we argued if VEGF inhibitors were going to work in any solid tumor, they should work in kidney cancer because of this link between VHL loss and upregulation of HIF and VEGF. And the good news is I think we're up to 8 or more VEGF inhibitors approved for the treatment of kidney cancer. So that's the good news. But the bad news is not every patient with kidney cancer will respond to these drugs, and even those that do will eventually progress. So I hope at least some of the students are thinking, well, wouldn't it be better than targeting a single HIF target gene to target HIF itself? And if you believe your own data, you should target HIF too. But the conventional wisdom, and students beware of conventional wisdom, the conventional wisdom was that HIF-2 was not druggable. It was a sequence-specific DNA-binding transcription factor. It wasn't thought to have a natural ligand-binding pocket, such as you would find in a steroid hormone receptor. So a number of pharmaceutical companies, certainly the classical pharmaceutical companies, said no way, no how. But one of our jobs in academia is to occasionally destroy conventional wisdom and dogma and paradigms. And fortunately here, the heroes, in my view, were Rick Bruick and Kevin Gardner, who were then at UT Southwestern. So they, first of all, they identified a potentially druggable pocket in the so-called PAS-B domain of HIF-2-alpha, which is what I'm showing you here. They then did high-throughput screens at UT Southwestern and identified chemicals that could bind to this pocket and in so doing induce an allosteric change in HIF-2-alpha such that it could no longer bind to ARNT, its partner, and hence no longer bind to DNA. Now, my drug hunter friends, and I can already see Nathaniel Gray making a face, my drug hunter friends tell me these chemicals were publication grade, but not pharmaceutical grade. These were never going to be given to an actual human being, but these were out-licensed to a company called Peloton that was later acquired by Merck, and now I do declare a financial conflict of interest. And the medicinal chemists at Peloton made these chemicals much more drug-like. They improved their potency, their specificity, their bioavailability, and they were kind enough to provide to us a tool compound, which at the time was called PT23099, I guess it still is. And this was like 1 or 2 atoms removed from what was their lead compound. And so our lab and the lab of my former postdoc, Jim Brugarolas, showed that this tool compound was highly active in various models of VHL defective kidney cancer. I also mentioned to the students at lunch, getting back to the reproducibility and robustness problem, that when I first submitted this paper, we had done this in 8 cell lines all VHL mutated, all overexpressing HIF2, and lo and behold, 4 of the cell lines were sensitive to the HIF2 inhibitor, and 4 were not. So I committed the sin of saying, this is what we saw. We don't understand it yet, but there appears to be some heterogeneity. And initially, the editor and the reviewer said, well, if you only understood this heterogeneity, if you only could tell us why these other 4 cell lines were resistant, we'll publish your paper. But without that, we're not going to publish your paper. But the advantage of having these gray hairs is I can call editors, and they sometimes respond to my phone calls. And I said, you know full well this is one of the things that contributes to the reproducibility and robustness problem, because a lot of scientists would then be tempted to say, let's sweep under the rug the 4 non-sensitive cell lines and tie a bow around the cell lines that are sensitive. Wouldn't it be better to tell the world you might see heterogeneity, and maybe we should start thinking about this? So fortunately, on this rare occasion, I carried the day. But I also want to show you one example, getting back to controls, of an experiment from our paper. So here's an experiment with a VHL mutated renal carcinoma cell line. You're looking at soft agar colonies, and we treated the cells with 0.2 or 2 micromolar of this HIF2 inhibitor, and you see decreased soft agar formation, soft agar colony formation. And the temptation is to say, eureka, high five, isn't this great? And you start writing the paper. But of course, first of all, I like to point out, this is the kind of experiment that gives cancer pharmacology and cancer biology a bad name, because this is a classical down assay. And I could have done this with Clorox bleach. I could have done this with formalin. And I would tell you at 10 micromolar, this chemical kills every cancer cell line we've ever looked at, whether it expresses HIF2 or not. So how would you know this is interesting and on target? So the gold standard way to do this is to do a rescue experiment, which Chin Cho in my lab did. So what I haven't told you yet is this cell line has wild-type HIF2 alpha. This is an isogenic control we generated with CRISPR, where there's a mutation in that pocket in HIF2 alpha that had been identified already by Bruick and Gardner that would prevent the drug from entering the pocket, but would otherwise leave HIF2 alpha intact. So now we could do this rescue. Now, in this case, we had the mutant delivered to us on a silver platter. We had to do the engineering, but basically it was delivered on a silver platter. But in other situations, we increasingly use CRISPR base editing technology to basically carpet bomb the putative target of the drug we're studying, and then to look for drug-resistant mutations. So eventually, an improved version of this called belzutifan went into phase 2 testing. So these are so-called swimmer's plots. Each horizontal bar is a patient on the trial. These are patients with advanced kidney cancer who had failed multiple standard of care agents, including VEGF inhibitors and immune checkpoint inhibitors. So for orientation, here's 1 year on therapy at the time of this analysis. The black arrow patients were on therapy doing well on the drug at the time of this analysis, and the yellow stars were patients who achieved a RECIST partial response. So based on this, these phase 2 data, the drug went into phase 3 testing, and I'll come back to that later. Okay, so now I'm going to share just a few unpublished vignettes. So the first is the work of Nitin Shiroli, a postdoc in my lab, and he wanted to know, with respect to the anti-proliferative effect of the HIF2 inhibitor, were some HIF2 target genes more important than others? So HIF2 actually regulates hundreds, if not thousands, of genes, potentially. But for this discussion, let's make believe there were only 4. So the idea would be in your kidney cancer with high HIF2, you have high expression of the ABCD genes, you come along with the HIF2 inhibitor, you silence them. And so his question was, what would happen if one by one I reactivated these various genes with CRISPR-A? Would any of them at least partially overcome the anti-proliferative effects of the HIF2 inhibitor? Now, I'm almost embarrassed to show this because, I don't have to worry about it because I took the slide out. I was going to show you a little bit more of the details of the CRISPR-A technology we were using at the time, but I have a feeling that this being Stanford, most people get the point of CRISPR-A. So what Nitin did was he took a renal carcinoma cell line that was highly HIF2 dependent. There's a nuclease dead Cas9 that's going to recruit a transcriptional activation domain. There's a little bit of other stuff, but not shown here. But basically, this is CRISPR-A. And then he introduced genome-wide CRISPR-A libraries. And then he treated the cells with DMSO or the HIF2 inhibitor, and then monitored guide abundance over time by next-gen sequencing. Now, one of the other reasons I like controls is often if you look in the controls, there's unanticipated useful information. So let's start with the cells that were treated with DMSO vehicle rather than the HIF2 inhibitor. So on the right are genes that, when activated, confer a fitness advantage. On the left are genes that, when activated, confer a fitness disadvantage. So right off the bat, if you look carefully, on the right is MYC, and there was already very good work from Celeste Simon and others that MYC and HIF2 cooperate with one another in kidney cancer. And we know they're cooperating because, as I'll tell you in a minute, when you treat with the HIF2 inhibitor, you no longer enrich for MYC. So this is specific, it's not non-specific. It's HIF2 and MYC doing a dance together to promote proliferation. But we were especially excited to see a gene that is probably less familiar to you called SQSTM1. And SQSTM1 is known to activate a transcription factor that most of us refer to as NRF2, but the formal name is NFE2L2. Now, the reason we got excited about this is the most common amplicon in kidney cancer occurs on chromosome 5q. And using what I would now consider horse and buggy technology, we decided that most arrows pointed to SQSTM1 being the most likely target of the amplicon. And now, with a completely different approach, namely CRISPR-A, we came to the same conclusion. So now let's look at the cells treated with the HIF2 inhibitor. So what we did was very simply to look bioinformatically for known and suspected HIF2 targets, and one that immediately caught our eye was cyclin D1. Now, cyclin D1, to people who think about kidney cancer, was interesting for a couple of reasons. First of all, in most tissues, in fact, virtually all other tissues beyond kidney cancer, HIF, if anything, indirectly silences cyclin D1. But for reasons we don't fully understand, in kidney cancer, HIF2 upregulates cyclin D1, which might be part of the answer for why VHL mutations predispose to kidney cancer, but not, for example, lung or breast cancer. Moreover, there's a human polymorphism within the HIF2 binding site at the cyclin D1 locus, and the polymorphism that's associated with tighter binding of HIF2 to the cyclin D1 locus is associated with an increased risk of kidney cancer. So that was pretty good. So we have cyclin D1 as a candidate of being a critical downstream target of HIF2. So just to show you how well this technology works, but again, maybe this is Coles to Newcastle, here are cells that have a control guide versus two different guides that hopefully are going to activate cyclin D1. The cells were or were not treated with the HIF2 inhibitor, so every place is a plus. You're treated with the HIF2 inhibitor. So as expected, we see downregulation of cyclin D1 because I just told you it's a HIF2 target gene in kidney cancer. But now we can maintain the expression of cyclin D1 with the CRISPR-A technology. So again, controls. Is this specific or non-specific? Here's another HIF2 target gene, NDRG1, and you can see it's unaffected. And then in low throughput experiments, cyclin D1 validated. I'm just going to show you the cell culture work, but the in vivo experiments worked as well. So here are the cells treated with DMSO. Here are the cells treated with the HIF2 inhibitor. And again, this is proliferation as a function of time. But now if you use CRISPR-A to maintain the expression of cyclin D1, the cells are actually now completely resistant to the HIF2 inhibitor. And so for this and many other reasons, clinical trials are planned, perhaps combining a HIF2 inhibitor with a CDK4/6 inhibitor. All right, so I'm now going to change gear and talk about another thing that's of interest, at least to people who think about kidney cancer, and that is kidney cancer has been suspected for decades of being highly immunogenic. So spontaneous regressions are actually well described. They're rare, but they're well described in the kidney cancer literature. This tumor has consistently high levels of T-cell infiltration. In the old days, these patients were sometimes treated with high doses of interleukin-2 or interferon alpha. And in fact, some of the patients treated with high-dose interleukin-2 were durable, complete responders, but it was such a toxic therapy, it was abandoned because these patients also often wound up in the ICU. And then they sometimes respond to immune checkpoint blockade. Now, we thought this was a great puzzle to work on because this is a fairly famous slide that might be familiar to many of you. I think it's the work of Gady Getz, where he's showing you the mutational burden on the y-axis and the tumor types on the x-axis. And I think many of you know that melanoma is sort of the 800-pound gorilla here, probably in part because of UV-induced mutagenesis. And it's thought that that's one of the reasons why melanomas are likely to respond to immune modulators. But again, the clear cell renal cell carcinoma, which is the form linked to VHL mutations, is smack dab in the middle. So why should kidney cancers be so immunogenic if they don't have such a high mutational burden? Now, We think a wildly underappreciated clinical experiment was the experiment that Richard Childs and colleagues did at the NIH 20 years ago, and he assures me this was done out of sheer desperation, and a lot of his colleagues thought he was absolutely crazy. But he treated 74 patients with advanced kidney cancer with allogeneic stem cell transplants as a source of sort of invigorated T cells. And about half the patients responded, and 10 to 12% had durable complete responses. And in one of the complete responders, he identified donor T cells that were recognizing a peptide on the cell surface of the tumor that was actually encoded by an endogenous retrovirus, which at the time he called HRVE. And he never found this retrovirus expressed in other cancers nor in any normal tissues. Moreover, there have been reports in the literature that the expression of endogenous retroviruses in kidney cancers at least correlates with the likelihood of responding to immune checkpoint blockade. Finally, the Childs group had gone even deeper. And again, I'm not sure why people didn't pay more attention to this, but they were able to show that this particular endogenous retrovirus was under the control of HIF2, and you're now experts on HIF2, which nicely explained why it would be upregulated in kidney cancer and not, for example, in other tissues. And parenthetically, I should tell you that HIF1 is expressed ubiquitously, whereas HIF2 is much more restricted in terms of its expression. So most cancers, for example, don't express HIF2. So another talented postdoc in the lab, Qin Jiang, wanted to know, was this patient described by Richard Childs, was that a one-off? Was that a unicorn, great for that patient, but never to be seen again? Or was it actually the tip of the iceberg? And so her hypotheses were that HIF drives the expression of multiple ERVs with the Child's ERV, which, and again, the nomenclature is absolutely god-awful here, but now it's called either ERV-E4 or it has a few other names, including 2256. So the question was, maybe there would be multiple ERVs regulated by HIF, and if that were the case, maybe some of these other ERVs would have the potential to be transcribed and translated and presented as HLA-bound peptides. And if that was the case, I think we could begin to understand perhaps why kidney cancers would be so immunogenic. So the first thing Qin did was she made 3 isogenic cell line pairs where HIF2 was high or HIF2 was low, so she could either reintroduce the VHL protein, she could treat with the HIF2 inhibitor, or she could eliminate HIF2 alpha with CRISPR, which you can do in these cells, at least in the short term, if the cells are in high serum. And then with the help of David Brown and Kathy Wu and others, we really focused on about 3,000 annotated ERVs in the genome that are relatively intact, where in your wildest imagination, you can imagine them having short open reading frames that might give rise to polypeptides. So we did RNA-seq using the 3 pairwise comparisons, and then looked at the abundance of these 3,000 ERVs. And I guess we have our first problem here from the transmission. Oh, here we go, good. So this is the Venn diagram of those 3 comparisons. And let's, for today's discussion, just be uber stringent and just focus on the 16 ERVs that scored in all 3 pairwise comparisons, although I'll tell you that many of the double hitters also validate in secondary assays. So again, for the students, it's always nice when you have an internal control, a sort of a reality test. Now, I wish I could say I planned it that way, but in this particular case, it was very gratifying to see that we rediscovered the Richard Childs ERV as being a HIF2 responsive ERV. And this other ERV, which has the trivial name of ERV 3.2, actually was featured in one of those 2 JCI papers I showed you. But now we have other ERVs as well that seem like they're HIF2 responsive in kidney cancer. So we decided, first of all, to ask whether some of these were really direct HIF2 targets. I'm not going to show you the ProSeq data, but here are some of the ChIP-seq data because they're kind of pretty. So here's the Richard Childs ERV shown here in the putative origin or direction of transcription. And for the students, corroboration is always your friend, right? So one way we did ChIP-seq was to use an antibody to HIF2 alpha for the ChIP-seq in cells where we did or did not eliminate HIF2 alpha. And there's a little ditzel there, but I'm not sure I would have taken that to the bank as a HIF binding site. But fortunately, Nitin Chiroli in the lab had already knocked in a flag HA tag into the endogenous HIF2 alpha locus of multiple renal carcinoma cell lines, including the 3 shown here. And now you can see with an anti-flag ChIP-seq very nicely this peak. And again, only of interest probably to the aficionados in the world. Childs had identified a putative HIF-2 binding site on what we'll now call the shoulder, but there's an even better one just north of it. But just for fun, here are two of the new ERVs that we discovered as being HIF-2 responsive, and I purposely picked ERVs where they're actually bookended by HIF-2 binding sites. Now, I'm sometimes asked, is there some teleological explanation for this, or is this just by chance? And I have to say my back-of-the-envelope calculations suggest this is probably by chance, but it doesn't mean it's not useful and fortuitous. But I don't want to go too far on a limb here and suggest there's some grand plan here as to why certain ERVs have neighboring HIF-2 binding sites. So then the next hypothesis, as I said, was that maybe some of these other ERVs are transcribed and translated. And so with the help of Carl Klausler and Steve Carr at the Broad Institute, we've done anti-HLA mass spec of cell lines, which I'm not sharing with you today, but as well as real tumors. So here are the data from 11 kidney tumors. And for 6 of these tumors, we had normal tissue. They did HLA mass spec and then looked specifically for peptides that could be encoded by our HIF-responsive ERVs. And suffice it to say, we have about 20 somewhat high confidence peptides here. You don't usually see the same peptide twice here, but I should point out these are all different HLA types. So that's part of the reason why there's this heterogeneity. Now, we went on to show that some of these peptides are actually immunogenic when given to humanized mice, but that seemed like kind of a low bar to me. So the question was, do peptides like this ever potentially contribute to the activity of immunotherapy in kidney cancer? And here's where we got extremely lucky. So I actually reached out to Richard Childs, who had done this clinical trial 20 years ago, and he still had in his freezer the samples from those patients. So your indirect cost at work. So they were frozen and stored and archived and annotated, and they were available for study. So he was able to initially send us the samples from 6 patients. And for one of the patients, the samples didn't survive thawing. For the other 2 patients, we didn't really see any evidence of reactivity. But in the other 3, we did. So these are busy slides. I apologize. So these are the data from so-called ELISpot assays, where we're trying to stimulate these T cells from these patients with either control peptides or peptides that we predicted might be present based on the work I just shared with you. So patient 1 is a patient who had a partial response, and statistically, several of these peptides did elicit immune responses. But I should also point out we had samples pre and post their allogeneic stem cell transplant. So in blue is pre-stem cell transplant, and red is after the stem cell transplant. So yes, there were some T cell clones that could react with these peptides, but they were present before and after the transplant. But there were 2 patients at the bottom here who had complete responses. And now you could see that in these patients who had complete responses, they actually had multiple T cell clones that could recognize some of these ERV-derived peptides. So that was exciting. But I have to tell you, I'm not an immunologist. I don't really like these ELISpot assays very much. If you really look at some of the controls, unfortunately, again, there are controls here, you can see the controls themselves are a little bit bouncy and noisy. Now, this is a log-based 2 scale, so my immunology colleagues tell me this is the real deal when it comes to ELISpot assays, but I was still kind of nervous. I didn't like how much noise there was. And so again, students, I told you a moment ago, corroboration is your best friend. So I learned from my colleague Glenn Drainoff that when he was head of immunology at Novartis, before he came back to the Dana-Farber, he had asked a company called T-Scan to see if they could understand why kidney cancer patients responding to immune checkpoint blockade responded. And their technology is based on technology developed by Steve Elledge. And the idea is the following. So you have a reporter cell that's been engineered so that if it encounters a T cell that sees something it likes, it goes bright. And so then what you do is you create libraries such that each reporter cell is going to express a different 90 amino acid polypeptide that then hopefully gets processed into HLA bound peptides. And fortunately for us, they had made a library that not only covered the human proteome, but covered many potential endogenous retroviruses. So they were kind enough to share some of their data with us. So they had done this on 7 patients with kidney cancer who had responded to immune checkpoint blockade. And they actually had taken so-called TILs, tumor-infiltrating lymphocytes, and then used this T-scan assay to see if they could uncover or identify the peptides that were likely being recognized by these TILs. So they had done this for 7 patients, but in 4 of the patients, either there was no VHL mutation or it was VHL indeterminate. And in none of those patients did they identify T cells recognizing ERV-derived peptides. But in the 3 patients who categorically had VHL mutations, for all 3 patients, they were able to identify TILs that recognized ERV-derived peptides, including in this patient, 2 peptides that were actually derived from the Richard Child's ERV, so that was sort of good here. They're using the 2256 nomenclature. 4700 was an ERV featured in one of those JCI papers I told you about as being potentially a predictive biomarker. And then in an example of the exception proving the rule, 1514, this ERV 1514, was not on our list. And so we went back and looked at 10 renal carcinoma cell lines at the expression of 1514, and in 4 of them, it was HIF2 responsive. So I think this is more evidence that maybe there is something to this notion that HIF2 is upregulating ERVs and that these are potentially immunogenic. Now, at this point, I had 2 choices. I might have guessed wrong. I was tempted to talk about our work on phenotypic screening for new molecular glue degraders, but you already have Nathaniel Gray and Jerry Crabtree, and lots of people who talk about this stuff probably pretty frequently. So I just wanted to end with one last kidney cancer story. So this is the work of Mohamed Abu Ramallah. So kidney cancer used to be referred to as the internist tumor because of all these very interesting paraneoplastic syndromes that sometimes cause patients to come to medical attention and see their internist. It might be fever of unknown origin, weight loss, polycythemia, et cetera. So for example, the erythrocytosis is almost certainly due to ectopic erythropoietin production. But 2 important paraneoplastic syndromes are cachexia, profound weight loss, and hypercalcemia. Now, Laura Stransky, when she was in my lab, noticed that if you looked at mice bearing xenografts with these OSRC2 cells I mentioned a moment ago, and you treated them with a HIF2 inhibitor, you dramatically prolonged survival. But this really, to our surprise, really wasn't explained by changes in tumor weight. Even the HIF2 inhibitor-treated animals had relatively large tumors at necropsy. But what was clear was that the vehicle-treated animals were developing profound cachexia, this is weight loss here, compared to the animals that got the HIF2 inhibitor. So somehow the HIF2 inhibitor was preventing the cachexia, and this seemed to be at least partially dissociated from changes in tumor mass. So we decided to use a technology developed by Alice Ting here at Stanford and also refined by Norbert Perman, where you have a promiscuous biotin ligase that's now targeted to the endoplasmic reticulum, and as a result, secreted proteins are then biotinylated, which you can then capture on streptavidin beads and identify by mass spec. So we did this with this cell line that causes cachexia, as well as a renal carcinoma cell line that doesn't cause cachexia. And we treated the cells with the HIF2 inhibitor or vehicle. So on the left are secreted proteins that are suppressed by the HIF2 inhibitor. So for example, just as a reality test, here's VEGF itself. And Peter Jackson and I were discussing sensitivity last night, but VEGF, I will point out, is present at picomolar amounts. So this is, maybe it's not perfect, but it was pretty damn good. But then we look for things that were seemingly secreted exclusively by the cachexia-inducing cell line, and that included, I know you can barely see this, PTHRP, parathyroid hormone-related protein. Now, my colleague Bruce Spiegelman had already linked PTHRP to cachexia in various lung cancer models. So again, we decided to look at this a little more deeply. There had been a report that PTHRP, and I should point out, technically speaking, the gene is PTHLH, was a HIF2 target. We've confirmed that with ProSeq. In fact, by ProSeq, it's amongst the most HIF2 responsive genes we've ever seen in kidney cancer. So even within 2 hours of treating with a HIF2 inhibitor, active transcription is completely or almost completely stopped. And then Mohamed, again, did sort of necessity and sufficiency experiments. So let's take the cachexia-inducing cell line. let's eliminate PTHRP using CRISPR. Now the animals live a normal lifespan. They no longer develop cachexia compared to the control guide cells. This is the change in body weight is quantified here. And again, this is not due to changes in tumor mass, as shown here at the time of necropsy. And as expected, this also correlates with the change in serum calcium. So again, the control guide animals have hypercalcemia, which is corrected when you eliminate PTHLH. Conversely, in sufficiency experiments, if you have a DOX-inducible version of PTHLH, which again is going to encode the PTHRP peptide, the animals rapidly die. They rapidly die because they undergo cachexia in front of your very eyes. Here's the body weight here. And again, this is not due to changes in tumor weight, but does correlate with changes in calcium. Now, does this happen in patients? So with the help of Eddie said and Tony Tuori here, you're looking at PTHRP ELISA experiments we did. And here you can see the upregulation in renal cell carcinoma compared to controls. Kidney cancer patients treated with the HIF-2 inhibitor belzutifan. And for comparison, patients treated with an immune checkpoint inhibitor versus a VEGF TKI. So first of all, you can see belzutifan very quickly normalizes PTHRP levels, so this looks like it's a pharmacodynamic effect. You lower serum calcium, as you would expect, and that's specific again because the immune checkpoint inhibitor and VEGF inhibitors don't do this. And finally, it improves body weight. The immune checkpoint inhibitors have no change, and if anything, patients on the VEGF TKIs tend to lose body weight because they don't tolerate the therapy very well. And finally, for corroboration, here's a completely different, it's the same pocket, so I shouldn't say completely different, but here's a chemically distinct HIF-2 inhibitor being developed by Nikang. And here what we did was we did ELISAs for PTHRP and then grouped the patients into quartiles. So G1 has the lowest PTHRP levels, G3 has the highest PTHRP levels, and G2 is in between. And what you're seeing here is the improvement in body weights in the patients who had the highest PTHRP levels. So in closing, I showed you this a moment ago. I told you the drug went into phase 3 testing. The phase 3 tests were positive, and the FDA approved the drug for the treatment of kidney cancer. But I thought since we're living in trying times, it would be nice to end on a particular high note. So a truism in medical oncology is that most drugs work better in a frontline setting than in very late lines of therapy. So we were able to convince Peloton and then Merck to treat 61 patients with von Hippel-Lindau disease who had measurable kidney tumors that had never been treated before. And these patients are often in surveillance programs where they get an MRI every 3 or 4 months, and only if a tumor changes its characteristics in an ominous way will they go to surgery. And this is in an attempt to delay or prevent the need for repeated surgery. So now you can see, again, here's 1 year on therapy. Everyone looks like, or almost everyone looks like they're doing well. If you prefer waterfall plots, here are the changes in the sizes of the indicator kidney tumor. So you can see everybody's tumor got at least somewhat smaller, including some patients who had partial or complete responses. You had to have a kidney tumor to get on the study, but you could have other tumors associated with VHL disease. You can see the hemangioblastomas shrinking. You can see these neuroendocrine tumors of the pancreas shrinking. And just to sort of emphasize further how life-changing this was for the patients, as I mentioned, these patients were in surveillance programs. So here's the 4 years of observation prior to going on a HIF-2 inhibitor. And every place you see a symbol, a triangle, a square, et cetera, the patient's going back to the OR to have some other tumor removed. And you can see when the HIF-2 inhibitor gets started, the frequency of the surgery goes down dramatically. And if you're a patient with this, and based just on the phase 2 data, the FDA approved the drug for the treatment of von Hippel-Lindau disease. And you can imagine if you've had von Hippel-Lindau disease, you've had sort of the sword of Damocles over your neck or head. It was just really gratifying to see some of the social media posts. Here's a VHL patient saying, "I never thought I'd see this day," and they're describing their tumors getting smaller, being stable, or disappearing altogether. And because everyone likes a vlog, and I do have the permission to show this patient's face, here's a vlog that was sent to me by one of the VHL patients.
Hey everybody, it's Justin, and I just wanted to give you a quick update. I am in a gondola right now in Taiwan. Over there is Taipei 101. The gondola is actually right by the Taipei Zoo. But I just wanted to give you a quick update and say I'm doing well. I'm enjoying my trip. if it wasn't for the PT2977 drug trial, I would have never been able to come out here and do what I'm doing right now. So I just want to thank Peloton, and I hope Merck will fast track this drug for VHL treatment. So if you guys are listening, hopefully you guys will put it on the market to help VHL. But yeah, keep watching these videos. I'll be making more, and I'll get better at it, and I have to get the angles right because it kind of looks fat, you know?
Yeah. So I love that, because at that age, that should be your biggest concern, is whether you look fat on your vlog and not what your next MRI is going to look like. So I think I'm going to close by just acknowledging that when I go to scientific meetings now, I feel like these two guys. Some of you may recognize this from the Muppets. So I'm entering the grumpy old man phase of my career, but I think there's a reason I'm entering the grumpy old man phase of my career. So I'm going to ask you to do something with me. So how many people, first of all, know who this is on the right?
I do.
Who is it?
Oh, it's, oh my God.
Well,
It's
no.
the professor, it's the professor and the kid who comes from.
Okay, so this is Mr. Peabody,
Mr. P, okay.
and this is Sherman.
Sure.
And they're standing in front of the Wayback Machine, the Wayback. So we're going to go into the Wayback Machine. I'm going to show you literally a slide I once showed at a scientific meeting when I was a postdoc, because this is what constituted a slide when I was a postdoc. And we could all look at it together. There were no animations. There weren't 15 panels with 4-point font, gratuitous Tisney plots and gratuitous U-maps and all sorts of things going. I mean, there was a thing. We all kind of looked at the thing together. And part of the fun, frankly, was we also showed all the data. Well, not this, okay. We actually showed what we saw. And half the fun was seeing if you saw something even the presenter didn't see. So, for example, if you're wondering, this is the equivalent of a Western blot, but rather than using an antibody to detect protein, I'm using a P32 labeled version of the retinoblastoma protein. And this was an extract from 293 T cells, and after transferring the extract to nitrocellulose, I cut adjacent strips. And so what you're actually seeing here is that RB is binding directly to E1A, which has this characteristic banding pattern. And in 2, I added a control peptide, and in 3, I actually added a short peptide from the HPV E7 protein that we suspected would bind to the same pocket within the RB protein. But if you're really squinting and looking really hard, there's a little faint doublet over here. This, in hindsight, was the first time I ever saw E2F. And this is what convinced me I could actually clone E2F using this technology with an expression cloning library. And then after you showed your data, you might have something that was no more complicated than this, which, for want of a better term, we'll call an idea that we wanted to share with one another and discuss. So I'm not trying to overwhelm you with like how many cell papers we had. I'm not trying to overwhelm you with how many panels I can fit in a figure. I'm just trying to share an idea, and we try to educate each other. And some people might say it's a bad idea. Some people might be a good idea. But to me, that's what a scientific meeting was. So I think we have to get back to using slides to educate and inform one another. I go to a lot of talks where even in the third row, I can barely see what I'm supposed to be. And I'm not even sure what I'm supposed to be looking at. In the back row, it's a complete, train wreck. And for those of you who don't think this matters, there actually are people who study how we educate one another. And so I would recommend to you Google death by PowerPoint or some variation thereof, because in this story they describe that actually the prescient warning for the challenger was on a PowerPoint slide, but it was such a horrifically bad slide buried with so much other nonsense, no one got the message that we better fix this before these poor people go up into space. So with that, I'll thank you very much for your attention, and I hope we have time for questions. And while we're doing questions, this is a QR code. You had a QR code?
Yes.
I have a QR code. This is a fantastic organization, the Environmental Voter Project, which uses analytics to find U.S. never voters who, had they voted, would have voted with the environment as their number one issue, using very robust, proven analytical models. And then they use proven behavioral science technologies to get these people to vote, including randomized controlled trials, so they know how many people they're actually generating. Not surprisingly, most of these people believe in science. You can guess how many of them vote. There are up to 1.5 million voters who never voted before this organization found them, and they think they can get to 5 million. And at that point, you have much more power than the NRA. And frankly, we start changing the kinds of elected officials we have in terms of how they do or don't approach science. So I think this is a worthwhile cause. Anyway, did I interrupt someone's question while I'm sorry, yes.
I was interested, you started the talk by mentioning that there's hundreds of thousands of targets of Hip signaling. And you also, you started the talk by mentioning that there's hundreds of thousands potential targets of Hip signaling, and you also showed some of those targets in that volcano plot. So I'm curious, how does this PT2399 compound avoid systemic hypoxic toxicity effects in the patients for whom you're showing therapeutic effects for?
Yeah. So I hope everyone can hear the question. So it is true that HIF potentially can activate at least hundreds, and maybe even on rare occasions, thousands of genes, especially if you include indirect effects. But many of those genes are quite context and tissue specific. For example, EPO, the ability to express EPO is restricted to certain cells. So nonetheless, your question is a good one. Now, again, I mentioned HIF2 expression is actually highly restricted. So right off the bat, we had the benefit that a lot of cells and tissues just don't express HIF2. So we don't have to worry about toxicity in those tissues. And then secondly, most of our tissues are not hypoxic at rest. So they're not, even if they had the potential to express HIF2 mRNA, they're not going to have a lot of HIF2 protein. So it turns out, now that the clinical experiment's been done, there are certain on-target toxicities, and some of them were predictable. So one of them is anemia due to suppression of erythropoietin and other erythropoietic genes, but that turns out to be manageable just by giving recombinant EPO. The more interesting one is some patients who were living at high altitude or relatively high altitude like Denver experienced, in most cases, asymptomatic hypoxemia, but hypoxemia nonetheless. And that seems to be a combination of the role of HIF2 in the carotid body, but perhaps more importantly in the pulmonary vasculature, and they experience worsening VQ mismatch. So that is another example of an on-target toxicity, but it turns out to also be quite manageable. And most drugs are drugs precisely because you can sort of titrate to the desired effect, and hopefully there's a therapeutic window. There appears to be a therapeutic window here.
That was great talk. So I have two questions. First is in your high-throughput screening, I cannot easily imagine what kind of cellular-based assay you perform for the high-throughput screening level.
Well, first of all, I wish I could say it was my screening. Again, that screen was actually done at UT Southwestern by Bruick and Gardner, and I think it was a modified version of a 2 hybrid looking at the interaction between HIF2 and ORNT. Okay. As you could imagine, various permutations of that. But if I recall correctly, and I will confess it's been a few years, it was a variation of a 2 hybrid.
You have one more question?
Yes, and then how you improve your the drug specificity after you find the preserved structure?
So the way, well, first of all, I mentioned we did the definitive experiment, which was to do the rescue with a drug-resistant mutant. You can also profile the chemical in various ways and see that it's pretty quiet, other than its effects on HIF2. And one thing you may be wondering, the analogous pocket in HIF1 is actually much smaller. So far, no one, and some people have tried, have been able to make an analogous HIF1 inhibitor. So even amongst the HIF family members, it's specific for HIF2.
In those rare patients who undergo spontaneous remissions, have you been able to study them at all for any immunological mechanisms?
Yeah, we'd love to. If anyone knows how to find these patients, I haven't found those patients yet. We consider ourselves very lucky that we found the samples we did for the patients treated.
It's usually those with lung metastases, if I remember.
That sounds about right, but I haven't revisited that in about 30 years, but that sounds about right. So if you have any of these people, let me know. And so now I'll do a medical student trick and answer a related question. You might have wondered, good question, can we actually upregulate HIF in other tumor types and turn on these ERVs? And the answer is yes. So if you take a pharmacological HIF stabilizer, we can turn on the ERVs in other tumor types.
We have a question online. This will be the last one because we're out of time, but this is from Dr. Sandy Srinivas, who says, I have several VHL patients on belzutifan doing really well. Can you comment on the resistance pathway and what we know about what's ahead for the patients who are on this drug who progress.
Great. So there have been, so far, I would say at the level of case reports, there have been case reports of patients who become resistant, either because they have a mutation very similar to the one we used, a mutation in the pocket of HIF2 alpha, or more intriguingly still, my former postdoc Jim Brugeros found a patient who had a mutation in the partner, ARNT, which increased the binding affinity so much that even the allosteric change didn't disrupt the complex. But I would say those are at the level of case reports. In cell culture, what we see is that some of the resistant cell lines actually have high levels of cyclin D2. And cyclin D2 seems to have taken over for cyclin D1. Whether that won't be important clinically yet, I don't know.